Hello everyone once again. So in this video we'll look at the full simple explanation of DNA replication. So first we'll note some few details. Like DNA occurs in a semi-conservative pattern. So what we mean by semi-conservative pattern is that the two DNA strands, by now we already know that DNA is a double helix structure, so the two DNA strands unwind and then they are used as template for synthesis of the new DNA strands. So there's usually an origin, a point in the DNA where we call the origin of replication. It's the place where the DNA unwinding takes place so that the synthesis of DNA can begin. And at this point, when the parent strands separate, they form what we call a replication bubble, as you can see in the diagram there. So we can locate the origin of replication and what we call a replication bubble. So there are enzymes. This is a summary of enzymes that are involved in DNA replication. So you can look at a video that I've done specifically for the enzymes to understand further. So for the enzymes, we have the helicase enzymes that is involved in the unwinding of the DNA double helix structure. Then we have the binding proteins which prevent the single-stranded, which maintain the, which prevent the single-stranded DNAs from now forming the double helix again. Then we have the primase. So once the DNAs have been unwound, or when they have separated and formed the single-stranded DNAs, there's an RNA primer that is usually added to now initiate the DNA synthesis process of the new template. So the primase enzymes is the one that adds the short RNA primers to the DNA. So once they add the short RNA primers, then the DNA polymerase adds the new template, the new nucleotides, which are the DNA nucleotides. So let's note the difference. The primase adds an RNA primer, and then now the DNA polymerase 3 adds nucleotides, that is DNA nucleotides, to the RNA primer. And then once that process is complete, now the RNA primase, the DNA polymerase 1 now removes the RNA primers and inserts the DNA primers to now form a complete DNA strand. And once that has happened, now the Okazaki fragments, you can check more on the Okazaki fragments in a video I did on specifically the Okazaki fragments. The Okazaki fragments are then joined together using the ligase to now seal the nicks that are formed in the sugar phosphate background, and then there you'll have your complete DNA. To process specifically, so we have what we call the leading strand. You can remember there's a video I've done on the structure of the DNA. So the DNA usually contains two strands. We know that the DNA has a double helix structure. For the two strands, we have one strand which is called the leading strand, and it runs from 5' to 3', and then we have the lagging strand which is from 3' to 5'. So the synthesis of the leading strand is usually continuous because the synthesis of both the leading and the lagging strand has to be in the 5' to 3' direction. So for the synthesis of the leading strand, it's usually continuous. But for the lagging strand, the synthesis occurs in the synthesis still occurs in the 5' to 3' direction, but because the template that is from 3' to 5' is the one that is used, the synthesis occurs in short, actually the short fragments are usually synthesized, and they are synthesized in the direction of 5' to 3', which leads to the formation of numerous Okazaki fragments. So as you can see at the top there, the overall direction of replication is on the right hand side. And of course now for the template that is from 3' to 5', the synthesis still has to occur in the 5' to 3' direction. That is the synthesis of the new DNA strand. So because of that, there are numerous primers will be added at several points. So the primers are denoted by the red color. So they're added, and then after that, the DNA polymerase 3 will add the primers. And then once that part is complete, again, the primer will be RNA primer will again be added in a different section, and then the DNA polymerase 3 will again add the nucleotide. So the process will continue and continue, and then it will lead to formation of numerous short fragments, which are called Okazaki fragments, as you can see in this diagram here. So here we have 3 Okazaki fragments that have been formed. And of course, the synthesis, as you can see, it's from 5 to 3' direction. But of course, the numerous short Okazaki fragments have been formed. That is in the lagging strand. So once the synthesis is complete, the DNA polymerase 1 will remove the RNA primers and replace them with the DNA nucleotides, because the entire new strand has to consist of the DNA nucleotides. But remember, the RNA primers are made of RNA nucleotides. Yeah, so the RNA nucleotides have to be removed, or the RNA primer has to be removed, so that the DNA primers can be used to fill those gaps. And the enzyme that is responsible for that task is the RNA polymerase 1. So once the RNA polymerase 1 removes the primers and fills those sections, the ligase enzyme now combines or fills the nicks of the sugar phosphate backbone to now form a continuous strand, a continuous new strand. So it combines those gaps formed during the synthesis process, because remember, the Okazaki fragments forms short fragments, or it consists of short strands. So these strands are then sealed together using the ligase enzyme to now form a continuous strand. And then this is just an overview of the replication fork, and it was obtained from the book. There, as you've seen, the copyrights are all reserved. So for the leading strand, we can see it's continuous, but for the lagging strand, we are seeing there are so many enzymes involved. So of course, there's the DNA polymerase 3, which now adds the fragments after the primer has been placed or added to the template. And then we have DNA polymerase 1, which now removes the RNA primers and adds the DNA template. And then now we have the ligase, which now seals the gaps to now form a continuous strand. And yeah, so the picture, the overall picture, is now termed as the replication fork. So thank you for listening. So in case you have any question, don't hesitate to ask in the comment section, and also you can like, subscribe, recommend to your friends. Yeah, thank you.